We will now continue with session 2, identification and development of novel surrogate endpoints for use in clinical development programs. In this session, our presenters and panelists will discuss the identification and the development of biomarkers as novel surrogate endpoints that could be used in clinical development programs. I would like to turn first to Dr. Charles Venditti, Senior Investigator in the National Human Genome Research Institute and the Director of the Organic Acid Research Section, and Dr. Oleg Shchelochkov, Associate Investigator at the National Human Genome Research Institute from the National Institutes of Health. They will be presenting together on methylmalonic acidemias and propionic acidemias. Welcome.
Thank you, Carrie. Next slide, please. Hi, my name is Oleg. I'm a clinical geneticist and physician scientist working with Dr. Venditti at his lab at NIH. Today, I would like to share with you one aspect of research conducted in the Venditti lab, focusing on the natural history of organic acidemias, and specifically how we leverage data sets to identify candidate biomarkers, validate them in a wide range of clinical and research contexts. My main focus today will be pharmacodynamic biomarkers, often referred to as response biomarkers, potentially suitable for future clinical trials. I also wanted to highlight that I use the term surrogate endpoint sparingly, in part because surrogate endpoint is context specific, and it requires discussion with FDA while designing clinical trials. Nevertheless, in my mind, response biomarkers tend to be attractive candidates to become candidate surrogate endpoints, so please keep that in mind. Next slide, please. Presented slides is a summary of hard work of many dedicated specialists working in Dr. Venditti's lab and our many colleagues at NIH and outside of NIH. Dr. Venditti is joining me today for this presentation and the follow-up panel discussion. Next slide. NIH offers a unique opportunity to conduct clinical, translational, and basic science research. Building 10 on the main NIH campus, shown here, is the world's largest clinical research hospital, whose infrastructure is optimally set up to conduct in-depth natural history studies of rare disorders. Clinical observations and resulting hypotheses made at the bedside while seeing patients can be readily tested using animal models housed on the NIH campus, as well as using in silico experiments on the NIH high-performance computing cluster called vivo. Next slide. Our study participants enrolled under the NIH Natural History protocols traveled to Bethesda, Maryland from the United States and internationally. A typical stay at the NIH Clinical Center lasts for 5 days. And during these 5 days, we conduct intense multidisciplinary clinical studies. We perform diagnostic imaging, collect DNA for genome and exome sequencing. We collect blood, urine, stool samples with subsequent biobanking. The data are then pulled into structured databases that we maintain internally. And we use data dictionaries, which are constantly updated, synchronized among different conditions, and harmonized with external data dictionaries such as Anvil, a project started at NHGRI. We also perform staged data releases in conjunction with publications that originate from our natural history studies. Next slide, please. Methylmalonic and propionic acidemia are severe life-threatening metabolic conditions that lack effective FDA-approved therapies. Solid organ transplantation is sometimes used in severe patients to reduce the number of metabolic crises. Hyperammonemic ketoacidotic episodes and chronic end organ damage are feared complications of this group of conditions. Multisystemic manifestations of organic acidemias require a comprehensive approach to subject evaluation. And one of the reasons for the presence of multi-organ damage stems from the nature of enzymes involved in this pathway. Next slide, please. Next slide. Thank you. Enzymes involved in propionate oxidation are ubiquitously expressed and feed carbon into the Krebs cycle. From the clinical perspective, deficiency of propionyl coenzyme A carboxylase results in propionic acidemia. Biochemically, it can be characterized by elevated 2-methyl citrate in body tissues, as well as propionylcarnitine, known as C3. The deficiency of the last enzyme in the pathway, methylmalonic coenzyme mutase, in methylmalonic acidemia, called so because of massive elevations of methylmalonic acid in body fluids. Finally, serum FGF21 and GDF15 can be elevated in the wide range of metabolic conditions that affect normal mitochondrial biology, although their role in MMA and PA is still incompletely understood. Next slide, please. The natural history study of methylmalonic acidemias at NIH began in Dr. Venditti's lab back in 2004. It represents the world's largest, most comprehensive prospective natural history study of this group of disorders. It encompasses more than 18 years of experience. 262 MMA patients have been enrolled so far, totaling more than 1,200 visits at NIH Clinical Center. Next slide, please. The natural history study of propionic acidemia at NIH began in late 2017, and today we have enrolled 44 participants. Each visit results in the collection of about 500 clinical imaging, laboratory, dietary parameters. A unique feature of both datasets is a subset of participants who underwent solid organ transplantation, such as liver, kidney, or combined liver-kidney transplants. Studying transplanted participants create an opportunity to evaluate candidate response biomarkers, their association with clinical meaningful outcomes, and their feasibility as surrogate endpoints in trials aiming to restore missing enzymes through interventions like gene therapy or mRNA therapy. Next slide, please. As we pursue novel genomic therapies, such as the AAV-mediated gene therapy, we were motivated to address the lack of FDA-approved response biomarkers and surrogate endpoints. It made sense to us to start with the studies of established diagnostic biomarkers, such as C3, methylmalonic acid, or 2-methyl citrate, which are used in newborn screening and diagnostic workups to confirm positive newborn screens. Next slide, please. A set of early experiments delivered promising results. One of the earliest observations made in Dr. Venditti's lab dates back to 2010. AAV-mediated gene therapy or gene transfer in mutase-deficient mice demonstrated the potential for this treatment approach. At the top, you can see a significant difference between a healthy heterozygous mouse and its untreated litter mate, an extremely rare affected mouse that survived until age 32 days. Treatment with an AAV vector encoding a healthy copy of the gene results in phenotypic correction of affected mice that persists through the first year of life and beyond. Next and next. Thank you. So as you can see on the one slide back, please. As you can see, mice that received that were treated with gene therapy are indistinguishable from their unaffected litter mates. Next slide. These phenotypic changes are accompanied by significant improvements in plasma level of methylmalonic acid. One important remark, though, is that although plasma MMA level in treated mice were significantly lower compared to the untreated animals, these levels in treated animals were still 100-fold higher than the normal range in wild-type mice. Next. And next. While plasma MMA performed well under carefully controlled lab conditions, our natural history studies conducted in parallel revealed potential shortcomings of the metabolic biomarkers. For example, plasma MMA showed significant intra and intersubject variability in our natural history study. Next. Or in the case of propionic acidemia, plasma 2-methyl citrate and propionylcarnitine, to establish diagnostic biomarkers, struggled to differentiate between two clinical states, before and after liver transplant. It posed a question whether these chemicals alone are robust enough to serve as single response biomarker in a future clinical trial. It prompted the lab to look for additional solutions. Next slide. And one more. And again. Thank you. One promising lead harnessed the power of non-radioactive tracer and flux biology in the form of 13C labeled propionate. Under in vivo conditions, propionate is quickly conjugated with coenzyme A to form propionyl-CoA. It then undergoes oxidation in the Krebs cycle to form labeled CO2, and this labeled CO2 can then be detected in breath. A standardized dose of GRAS grade 113C propionate has been tested both in mice and then later in human study subjects under various physiological conditions with similarly revealing results. Next. And one more time. Thank you. So as you can see, this is these slides demonstrate the clinical setup for these studies at the clinical center and NIH. And it involves a collection of breath samples shown in the lower left photograph. Next. This graph demonstrates how affected mice show significant increase in the rate of propionate oxidation after AAV-mediated gene therapy. Next slide. Similarly, MMA patients who underwent combined liver kidney transplantation demonstrated significant improvement in the rate of propionate oxidation. Next. As you can see on this slide, patients who underwent liver kidney transplant are nearly indistinguishable from healthy controls. On the other hand, patients who received only kidney transplant were experienced only marginal improvements in their oxidation rates. Next. You can read the details of this study in the paper that is quoted here. Next slide. With the arrival of the NIH-PA cohort, we hypothesized that diagnostic biomarkers of propionic acidemia, C3 and 2-methyl citrate, as well as the in vivo oxidation of propionate will correlate with clinical outcomes of propionic acidemia. In addition, we sought to establish an analytical framework that would allow us to not only critically examine the most obvious candidates for response biomarkers, but also scale it up by evaluating the entire set of biomarkers, about 500 of them that we collected during the natural history study of propionic acidemia at NIH. You can refer to the paper cited here for additional details about how our machine learning approach was used to prioritize and select biomarkers. Next slide. Consistent with our hypothesis, in vivo oxidation of propionate in PA showed excellent correlation with established diagnostic biomarkers, C3 and 2-methyl citrate. But it also correlated with clinical outcomes such as full-scale IQ and linear growth, to list a few of them. Next slide. In addition, propionate oxidation demonstrated its resilience over a wide range of renal conditions. This was in contrast to C3 or 2-methyl citrate, which showed significant dependence on the renal clearance. It's a common problem in organic acidemia, where chronic kidney disease is a common complication. Next slide. Furthermore, in vivo labeled propionate oxidation shows significant improvement and even complete correction in liver transplanted propionic acidemia subjects, as well as strong intrasubject reproducibility. Next slide. In addition to identifying propionate oxidation as a potential response biomarker suitable for clinical trials, we identified novel candidate response biomarkers using the machine learning paradigm that was described in the paper that I cited before. And some of these biomarkers that were prioritized by the machine learning algorithms are shown here: plasma FGF21, alanine to serine ratio, and GDF15. At least 2 of them have now been shown to correlate with outcomes in independent cohorts of patients and in vitro studies supporting the use of our machine learning approach. Next slide. In summary, development of response biomarkers in organic acidemias can benefit from complementary observations made in human subjects, model organisms, and in silico statistical modeling. Studies of the liver transplant cohort in participants provided us with unique opportunities to test candidate response biomarkers that are designed to detect interventions to gene therapy replacement trials And we hope that our machine learning approach will inspire other investigators to adopt this algorithm to mine rare diseases data sets and enable future clinical trials. Thank you for your time.